Now we're going to do a little bit of a deeper dive into some specifics with soil health indicators. And we're going to get into specifically biological indicators of biological activity. Recall that there are a lot of different indicators that are out there, different types of measurements that we can make that are vetted measurements, published measurements, or published protocols, for example. Things that are looking at soil biological indicators. So again, the screening process and figuring out which ones are amenable for kind of rapid commercial soil health quantification. At this point, we should understand that there has been a historical focus on testing soil chemistry as a major kind of component of fertility in soil, and thinking about the role that things like pH, CEC, extractable potassium, phosphorus, calcium, magnesium all play in soil health. But what about soil biology? What are the things that we can measure that specifically deal with soil biology and soil biological activity? Well, we can think about microbial or microbial/biological measurements, and there's a number of approaches or different philosophies or concepts that we can think about quantifying. Okay, so one of the approaches is asking the question, how many are there? And we can look at things like plate counts for actually estimating a total, like a CFU, a colony forming unit. So that gives us some actual quantification. We can measure microbial biomass in the soil, which gives us a measurement of the total biomass of the microbes in the soil. We could do things like microarthropod counts or nematode counts, for example, and get some idea of like quantifying or totaling or tallying how many of these things are there. Another approach is asking who is there, who are the actual critters that are in our soil. And for that we can do things like measurements of species richness. That's a measurement of total species number, or often in the case of microbes and in the case of bacteriology or microbial ecology, we are looking at what we would call an OTU, an operational taxonomic unit, which is essentially a proxy for, can be considered the same as, analogous to, it's not really the same thing, but analogous to, say, individual species or different genera of bacteria or archaea, for example. We can look at something like diversity, so looking at the distribution of and evenness of different critters that are in the soil, right? So this whole idea of like biodiversity and looking at, there's lots of different microbially, a lot of different methods to get at this sort of thing. We can actually do a like a nucleic acid sequencing, where we actually sequence DNA, and then we find out, we reference that with a known database of microbial DNA, and then we can figure out who's actually there and perhaps get some idea of relative abundance of those organisms. So, all these kind of methods go back to who is there, right? We can ask things like who is there and in what proportions, and so we can look at community composition, either with something called phospholipid fatty acids, or PLFAs for short, DNA profiling, fingerprinting, or sequencing. These are all different techniques with strengths and weaknesses for asking who is there in what proportion. So kind of a fourth category of types of questions we can ask are things like, how active are they, or what are they doing? So here we're not looking at quantifying individual critters per se, or who is doing it, but we're actually measuring rates of processes. So things like enzyme activity, decomposition, or rates of a process. So an example of this could be like nitrification. So we take a soil sample and we measure production of nitrate, for example, or we measure production of ammonium, or we will look at a added specific substrate and see how fast that substrate is broken down. So these are all examples of activity. Okay, obviously depends on what your question is and what you're trying to get at, but we hear a lot of farmers say, oh, well, biology is really important. We really need to start measuring biology. Well, there's a lot of complexity and nuance in a statement like that. And we need to understand that there's a lot of different approaches. And some of these methods are very expensive, very time consuming. And when you get the results, it's really unclear what the implications are for, the results you see. So there's a lot of work to be done in this. And then we can also think, when we talk about biological or microbial activity, should the focus, where should we place the focus? We can think of these soil food webs, and we can think of, bacteria as kind of like at the base of that food web, perhaps. Fungi might also be at the base. We have things like nematodes, things like microarthropods, macroarthropods, and then we've got, other foragers. What should we target, and how should we do it? So these are all questions that need to be considered when thinking of this. I'm going to talk, and this is like most things, this will be a little bit impose my bias. We've already talked about our active organic matter, and thinking of that active pool. This is something that we've spent a lot of time in my lab looking at that active fraction, because it's we believe this as an indicator, as an indicator and an integrator of the soil biology and the soil chemistry. It's a bit of a biochemistry analysis or approach, but it really is reflective of gross processes from the soil food web. Okay, so what we've done. We've taken 3 tests that we really like and we feel confident in, and we've done a lot of work in this over the last 3, 4, 5 years, and we're still trying to figure a lot of this out. So this is still this talk is very much a work in progress. But we've got respiration as our first measurement, as a measurement of total microbial activity. I'm going to get into each one of these in a little bit. We've got active carbon, which is a measurement of the labile carbon pool. And we've got a soil protein as a measurement of an organic nitrogen pool. So collectively, we think that these measurements are related, but not redundant, and they're complementary in terms of the information that they provide. Okay. The first thing I'll highlight is this active carbon or available carbon. Active carbon is a rapid and inexpensive method to measure organic matter that is oxidized by potassium permanganate. Okay, so it's a potassium permanganate solution. It uses 0.02 molar potassium permanganate, and what happens is that starts as this. You'll see this really brilliant purple oxidant. It's a general oxidant. When it gets in contact with some available or chemically active carbon, it reacts with that, and then that permanganate gets consumed, and the color goes from a bright purple to kind of it will go all the way to clear, if you have enough of the material, enough of that feedstock. So in the literature, from a lay perspective, we call it active carbon. From a scientific perspective, we call it permanganate oxidizable carbon, just to be a little bit more precise. The measurement is outlined here. It's 2.5 grams of soil, 20 mils of 0.02 molar potassium permanganate. We shake, we put that in a centrifuge tube, 50 mil centrifuge tube. We shake for 2 minutes. We settle for 10. Then we dilute it down. It's a 1/100 dilution, so we take a 0.5 ml of the supernate from this reaction and then add 49.5 mils of water. We mix that and we read it on the spectrophotometer, okay? So again, as you know, there's more of this active carbon or POX, that permanganate, this dark, bright, brilliant purple solution, will react with that, consume that, and then as that permanganate is consumed, it essentially turns to clear. It loses its pigmentation. So then we take and we read this on a spectrophotometer and sees and that spectrophotometer will quantify how much light passes through this. So the darker the tube, there's less of active carbon. The lighter the tube, there's more active carbon. Okay, so it's kind of maybe a little counterintuitive. It's a little bit of an inverse relationship there. POX reflects a processed but available pool of organic matter. We know it's biologically active, it's sensitive to management. It represents a small percentage, again 1 to 4% of the total organic carbon, okay? So we have done a fair amount of work with this measurement, and just time and time again, it seems to be a very good indicator. It's sensitive to management, it's repeatable. There's a lot of great benefits to this, and it's incredibly simple, as you can see. There's really no, we run many hundred of these in a day in our lab. You don't need any fancy equipment or anything. It's a relatively straightforward test. Okay, the second is soil respiration. Respiration is exactly what you'd think it is. In context, it's really just measuring the soil microbes breathing. It's like the breath of the microbe. So it's how much CO2 is evolved or is released in an incubation, okay? So technically it integrates the size of a labile pool and the metabolic potential of that microbial community to decompose substrate. So this is a measurement that's been around for quite a while, but it's been simplified in maybe last 15, 20 years. And there's a number of different ways that we do it. Some of it's done on field moist soils, that is soils that are sampled in the field and they already have moisture in them. But by far the more common way that it's done is that you sample soil, you sieve or mix that soil to some degree, various ways. You air dry that soil, and then you add water. And when you add the water, you wake the microbes back up. You incubate it for usually 1 day, 24 hours. Sometimes it's up to 3 days. And then you measure how much CO2 is emitted from that soil. And that can be done directly by using a syringe and measuring the concentration of gas, or you can trap that CO2 in some various things, okay, and then quantify a pH change. And so Solvita is the commercial implementation of this method. Solvita just took a method and essentially developed this kind of this implementation where this is a CO2 trap. As that CO2 diffuses from the soil into this paddle, that paddle will change colors with an indicator dye. So it's really the CO2 acidifying this paddle, and that is what changes the colors. You can read that with just a color chart, or they've got these digital readers that will do a little bit more precise job. So this is something that you can buy and just run in a commercial lab. You can run it as a farmer. You can there's a number of different things, and this can be done in the field as well. The way we do it in our lab is we will usually add 10 to 20 grams of soil. We'll add a predetermined amount of water, and this is not particularly easy to figure out, and there's a lot we could say about that, but we really need to, adding the amount of water, the right amount of water, knowing what that right amount of water to add, it can be challenging. We seal this in a jar, so either these like pint or up to quart sized mason jars, or sometimes 50 ml centrifuge tubes right here. We add water, immediately seal it, incubate it for a day, and then we simply a little syringe here, and the septa, you push the syringe in the septa, you withdraw some of the air in there, and then you inject it into a instrument, an infrared gas analyzer, and it reads CO2 concentration. This little box, it's in a little infrared that reads concentration of CO2. So you get concentration of CO2, and it's a very simple test. And this is a test that farmers can relate to, because it's like, oh, the more life I have in my soil, the more it breathes, the healthier it is. It just makes sense. I'll revisit this idea of stabilization versus mineralization, okay? Stabilization being kind of locking up nutrients and carbon, building organic matter, and mineralization being this process of releasing fire. Are you building a wood pile, or are you making a fire and releasing it? Both of these things happen simultaneously, but if we have a teeny bit more stabilization than mineralization, year after year, we're going to be building organic matter and vice versa, right? So we've used this, and we think this is a good, valuable framework that we've developed in our lab to look at the relationship between active carbon, or POX, and respiration, mineralizable carbon. So, we graph these separately or on different axes, and in this scenario where we have low respiration but high active carbon, we've got greater stabilization. Conversely, if we've got high respiration values on a soil and low active carbon values, we believe we're having greater mineralization. And so, obviously, what we want the best of both worlds. We want to target high stabilization process and great turnover. So we want a lot going into the soil, but we also want that getting decomposed and have this very active, rapidly cycled organic matter. That's what's, the kind of bread and butter from a nutrient perspective of a very healthy soil. And so we've looked at this, and we, have basically found a very consistent relationship in all the data sets that we've looked at to this point. So it's very promising, where if we take the same, say this is a long-term experiment for Michigan, and we look at treatments that had composts with them, or so here's a treatment with a compost and with a diverse crop rotation. Here's no compost but have that diverse crop rotation. And then these are both continuous corn with and without compost. And they basically fall on, you graph these together, and they fall on areas where we think when we're having this diverse crop rotation, we're going to be in a period of organic matter building. And when we're in just continuous corn, we're going to have more respiration, more essentially mineralization release, because there's a lot of fertilizer that's going in with corn after corn after corn, etc. So there's some complexity here that I'm not going to get into all the details, but a few years ago we published a paper looking at this relationship between active carbon and respiration and how management influences these things. So management disproportionately influenced active carbon. The management practices that did that were things like conservation tillage and compost. These are practices that we know are very effective at kind of storing organic matter, building organic matter. And then management practices that disproportionately influence respiration values were things like tillage, manure application, and cover crops, particularly leguminous cover crops. So, when we add, for example, compost or have a diverse crop rotation, often we get increases in both active carbon and respiration. But figuring out which one is getting tweaked a little bit more, which one is getting more influence, that's what the result of this work was. And I won't get into all this, but suffice it to say that we found very consistent relationships, and we think this framework is a helpful framework to educate farmers to think about like what state their soil is in, and are they in a state of active matter, an early indicator of organic matter building or an early indicator of organic matter decline. I ask growers, what do you want? Do you want to build organic matter, or do you want organic matter to get released to feed your crops? And, of course the answer is, well, both. From a long-term perspective, we want to build organic matter. But from a short-term perspective, we've got to feed the crops and have good nutrient turnover to have productive crops. And so, this is a tool, potential tool to help them manage and understand what's happening in their soils in a particular field. Okay. All right. So the last piece is the soil protein, and we've talked about how do we quantify different ways that we quantify nitrogen pools, and we talked about KCl extractions. Ammonium and nitrate is probably the most common nitrogen test. But then there's also a labile organic pool of nitrogen, different ways that we can quantify that, different methods to do that. Okay, and this again goes back to this idea that total nitrogen is a very important measurement, but changes very slowly over time. So it might take years or even decades to see changes in total nitrogen, depending on a management practice. But we want something that's not too ephemeral, not doesn't bounce all over the place, but something that is a little bit more sensitive to management. That would be a good soil health indicator. So soil nitrogen is or soil protein rather, is one of those possibilities. Okay, there's again this split of inorganic versus organic, and organic often has incubations or might look at analysis called amino sugar. We're not going to get into this, but these are other different analyses that are out there. But we've really focused a lot on soil protein as a measurement. The method itself is 3 grams of soil in a sodium citrate buffer solution. You shake for just a few minutes, autoclave it for 30 minutes. So it's that autoclaving that it's a high temperature and pressure. It pulls that protein out of from the soil out into solution. We cool it from the autoclave, shake for 5 minutes, centrifuge, and then we transfer it to a plate. And then we incubate it for an hour and let this protein develop colorimetrically. So it's a colorimetric similar to the active carbon. It measures absorbance and transmittance of light through that plate. And we can relate these with standards, and we can quantify with very good precision how much protein is in that soil. Okay, so this is a way that we do this, and again, it's rapid and it's high throughput, and we still have a fair amount of work to do to figure out exactly what the soil protein functionally reflects and how it can be used to help with management. But the long-term goal here is that we can that we're going to be able to, develop a good soil nitrogen test. This is kind of a holy grail in soil science, particularly in agronomy, but just trying to find a good nitrogen test that's out there. Okay. All right. So again, the 3 of these together: respiration, active carbon, and soil nitrogen, coming together to kind of form complementary but not redundant tests or indicators. I'll just end by mentioning a couple different indicators on the horizon or methods on the horizon. The first one we've been working quite a bit with is this idea of infrared spectroscopy. There's lots of different forms out here, but for soils, there's near infrared, there's mid infrared, there's visible infrared. We've been mid infrared happens to be a really they're all good in their own right, and they all have pluses and minuses, but we've been working a lot with the mid-infrared. This is a non-destructive, very rapid test. We've got a auto sampler, so we load a plate up, we put it in, and it reads each one of these wells. The way it works is it shoots energy, energy, and then it measures at that soil, measures what's reflected, what's absorbed, and you get essentially a spectra off that soil. The beauty of this technology and methodology is that with this and with a lot of fancy statistical modeling, we're able with very high degree of accuracy to predict soil properties, a whole host of soil properties, things like total carbon, nitrogen, texture, things like active carbon, likely things like soil protein. So, with one, say, 30-second or 2-minute scan on a soil, we might be able to essentially, with enough background work in developing these calibration curves, be able to predict and predict a wide range of soil properties and make soil health testing very, very affordable, very inexpensive to do this. So that's, coming at it from a very applied, practical perspective of if we want farmers to soil health test and manage things like their biology, manage things like the physical structure, can we make soil testing for that really cheap? And this is a great way that we might be able to do that, okay? We can even take it one step farther and say, well, why even measure? This is a bit tongue-in-cheek to say this, but like, why even sample soil? Can't we just have sensors that measure soil properties and relay those back? So this is one example. Precision Planter is a John Deere acquired company. They do a lot of great things all around very precise placement of seed, very consistent, precise placement for better stands and better agronomy essentially. One of the things they have is a product called a Smart Firmer, which is this is a seed firmer. So the seed is placed in front, and then this is drug over to basically firm that seed into the seed furrow before the closing wheels will close up that crease that row unit is made. This is a sensor that's drugged through the soil, or it's attached to that seed firmer, and it has sensors on it that measure soil moisture, soil temperature, and organic matter. So those are the three properties that it spits out, and integrated into a lot of precision planting framework. They've got software, and everything's GPS referenced, so you can go through this. These things are on, of course, individual row units, and you can get a map. You can essentially plant your field and get a map of moisture, temperature, and organic matter to boot, right? So you can go, after you run through the field and plant everything, you can look at that and use that map as a management tool for moving forward. So something like this, although I would caution there's a lot of work that has to be done still to calibrate this and make sure it's actually a good scientifically valid technique or instrument. A lot of times companies are so quick to push these out into the market without any real vetting, or they do internal testing, but it's unclear how much we can trust that sort of thing. But this type of technology is very exciting, I think, and where we continually want to look in terms of making things very data rich, spatially explicit, and very cheap, right? I mean, it couldn't be cheaper. You just have to buy these row units once, and then you're done, and you can use this over year after year after year. So, okay, so soil biological measurement, and this one, again, for this organic matter piece, that's why I threw it in here. But I just think it's kind of interesting to think about some of these things that are on the horizon.